Today's topic is diphtheria. So it is a 15-mark question. It's generally given as a clinical question. So when it is given as a clinical question, the keywords in the clinical question to diagnose diphtheria are. So in the question we can observe the words like bullneck and gray-white membrane on the tonsil, and generally it is present in the children, and the children presents with fever. The child have foul breath, and throat swab when sent for the examination shows gram-positive bacilli. So the keywords here are bullneck appearance and the gray-white membrane on the tonsil, and also foul breath. So when you observe these points in the question, you have to diagnose that the question they're asking is diphtheria, and you have to write about Corynebacterium diphtheria. So, what is diphtheria caused? What causes diphtheria? So, diphtheria is caused by Corynebacterium diphtheria, which is what is Corynebacterium diphtheria? It is a gram-positive bacteria. Corynebacterium diphtheria is a gram-positive bacteria. What it is a club-shaped bacteria. Club-shaped means it have rod and swelling at the top. So it have Chinese letter arrangement. It has metachromatic granules. The metachromatic granules you can observe in this diagram there with rod and a swelling on the top is metachromatic granules. They are generally used for the storage. So, what are the virulence factors of diphtheria? How diphtheria causes the disease? So, the diphtheria, Corynebacterium diphtheria generally releases a toxin called diphtheria toxin, which is DT, which is the primary virulence factor. So, diphtheria toxin is a polypeptide chain. It is a polypeptide chain, which means it is made up of protein. The polypeptide chain contains two fragments, the fragment A and fragment B. So, A is the active fragment, B is the binding fragment. Okay, so by what mechanism diphtheria? What is the mechanism of diphtheria toxin? How it causes the disease? So, mainly diphtheria toxin enters the cell and prevents the protein synthesis. Due to its main role is to prevent the protein synthesis. How it prevents the protein synthesis? So the following steps are binding. First step is binding. The toxin binds to host receptors like epidermal growth factor on the cell surface. After binding, the cell engulfs the toxin into the endosome, which is called endocytosis. After engulfing, the toxin splits into two fragments, fragment A and fragment B. The step is called fragmentation. After getting fragmented into A and B, the B fragment create holes in the endosome membrane, so which leads to formation of a hole in the endosome. To that endosome, fragment A is released into the cell cytoplasm. So fragment A helps in fragment B to get released into the cell cytoplasm. Here in the cell cytoplasm, the main step occurs, which is ADP ribosylation. Fragment A is the active fragment causes ADP ribosylation of elongation factor 2, which is elongation factor 2 is plays an important role in the translation step of protein synthesis. Due to ADP ribosylation of elongation factor 2, inhibition of elongation factor 2 occurs, which leads to inhibition of translation step of the protein. So there is no protein synthesis. So through this process, when the examiner asks the mechanism and pathogenesis, you have to write these points. Next, what are the factors regulating toxin production? How the toxin is released? Diphtheria toxin is mainly produced due to beta-corniphage, which is a bacteriophage, which carries tox gene. Corynebacterium diphtheria remains toxigenic as long as these phages are present inside the bacilli. As long as beta-corniphage is present in the bacilli, which contains tox gene which is coded for the diphtheria toxin, the diphtheria toxin remains toxigenic. So, what are the clinical manifestations of diphtheria? So, the diphtheria is toxin mediated. It is directly not caused by the organism, it is caused due to the toxin released by the diphtheria. The bacilli are non-invasive, means they are present at the location where they are produced generally, like local sites like pharynx. So when they are secreted into the bloodstream, they are spread via bloodstream to various organs. Coming to the main clinical manifestation, respiratory diphtheria. The respiratory diphtheria is most common form of diphtheria. It is mainly affects the tonsil and the pharynx are the most common sites. Incubation period is about 3 to 4 days. Diphtheria toxin elicits inflammatory response. Due to the inflammatory response, which leads to necrosis of the epithelium, due to necrosis of epithelium leads to formation of mucosal ulcers. The mucosal ulcers are lined by tough leathery grayish-white pseudomembrane. This is the key point. So, the pseudomembrane composed of inner band of fibrin, and it is surrounded by neutrophils, RBC, and bacteria on the outside. So, what are the keywords? Pseudomembrane and bull-neck appearance. Extension of the pseudomembrane in severe cases may occur. So, the pseudomembrane extends into the larynx and bronchial airways, which may result in fatal airway obstruction, leading to asphyxia. So, this mandates immediate tracheostomy. Next, coming to bull neck appearance. The bull neck appearance is due to massive tonsillar swelling and neck edema, with the patient generally presents with foul breath, thick speech, and noisy breathing due to bull neck appearance. So, what is the laboratory diagnosis of diphtheria? So, what specimen should be collected? The specimen to be collected is the throat swab and a portion of pseudomembrane. So, coming to the direct smear, which includes gram stain, the gram stain shows club-shaped gram-positive bacilli Chinese letter arrangement. So, Albert stain is important here. The Albert stain, when done to this, the green bacilli with bluish black metachromatic granules is observed like this. So, coming to next, culture media, which includes enrichment medium, are blood agar and chocolate agar. The selective medium is important. The selective medium is potassium tellurite agar, which produces black colonies. So, other methods for to by using the PCR, we can detect the tox gene. By using the ELISA, we can detect the toxin. These points are also important. So this is all about diphtheria. So thanks for watching.